In this video, we'll be talking about the nucleic acids, and by nucleic acids, I mean DNA and RNA. DNA and RNA are both large, complex molecules, and they are polymers of molecules called nucleotides. DNA is double-stranded, so this is DNA here. You can see there's one strand kind of wrapping around that way and a second strand on the other side. So if you look at DNA, it's kind of got the shape of like if you took a rope ladder shaped like that and you twisted that around a central pole. That would give you the rough structure of DNA. RNA over here is single-stranded, so there's only one strand. DNA and RNA also differ in function. DNA carries the genetic information from generation to generation, so you share half your DNA with your mother, your biological mother, and half your DNA with your biological father. Kind of an amalgam of the two because you got DNA from both parents. RNA helps take that genetic information that's in DNA and translate it into proteins. So we'll talk about this more when we talk about how cells function, but for now understand that RNA is an intermediate step. RNA is an essential step in taking the information in DNA and using that information to make proteins that actually carry out the work of cells. Okay, so now that we've got some rough idea of the large structure of DNA and RNA, let's talk about the monomers. Let's talk about what DNA and RNA are made up of. Nucleotides are complex molecules themselves that consist of a central sugar group, a phosphate group coming off of that, and a nitrogenous base. The nucleotide structure is a little bit different depending on whether you're talking about DNA or RNA, particularly the sugar. If you're talking about DNA, that sugar is deoxyribose, one of the pentose sugars. If you're talking about RNA, in an RNA nucleotide, the sugar is ribose. It's right there in the name. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Deoxyribo because it's deoxyribose. Ribo because it's the ribose sugar. There are 5 different kinds of nitrogenous bases. You have adenine, you have guanine, you have cytosine, thymine, and last but not least, you have uracil. Adenine and guanine are what are called purine bases. That means that they have a 2-carbon ring structure, like illustrated here. Cytosine, thymine, and uracil are the pyrimidines. They just have a slightly different structure in their nitrogenous base. They only have 1 carbon ring. Adenine, guanine, and cytosine are used by both DNA and RNA. But thymine is only used by DNA, and uracil is only used by RNA. So DNA, in addition to the differences in the sugar, DNA uses A's, C's, G's, and T's, and RNA uses A's, C's, G's, and uracil in place of thymine. Now, nucleotides are the monomers of DNA and RNA. So when you attach nucleotides together, you get an alternating backbone of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Just one more for kicks. And the nitrogenous base sticks off of the sugar and for the nitrogenous base. Right? So for a single-stranded RNA, that's what you have. This is the sum total of RNA. It's just a single strand of nucleotides strung together with the nitrogenous base kind of hanging out there. But when you make a double-stranded molecule, DNA, you still have that sugar-phosphate backbone, with the nitrogenous bases sticking off the side. And the nitrogenous bases, where they meet in the middle, they have hydrogen bonds that hold them loosely together. They aren't permanently attached, but they are attracted to each other. And even though each individual nitrogenous base interactions are pretty weak, when you have, hundreds of thousands of them lined up along a single DNA molecule, the collective cohesion between these two strands is pretty strong. And they're stuck together because of these bonds between nitrogenous bases. So if you think back to our image of DNA as kind of a rope ladder, each rung of the ladder is formed by two nitrogenous bases, and it's always a purine paired to a pyrimidine. This pairing of nitrogenous bases is not random. It is very, very specific. You always have G paired with C, guanine with cytosine, and you always have adenine paired with either thymine or paired with uracil, depending on whether the partner is DNA or RNA. Base pairing is what holds the two strands of DNA together. And you also have base pairing when RNA is being made. So in the process of making RNA, you have a brief moment where you have the DNA strand paired up with the RNA strand. And this is a situation where you might have adenine in DNA paired up with a uracil in RNA. Now we'll come back later in the quarter to how proteins are actually made, but it's the sequence of the nucleotides that can be read to make proteins. So if you were reading DNA, if you've ever heard anyone talk about DNA sequencing, what they're talking about is the sequence of the nucleotides, the A's, C's, G's, and T's, right? And it's the sequence of nucleotides that gets transcribed into RNA, into kind of an RNA copy, and that sequence of nucleotides in RNA that's actually read to determine the sequence of amino acids in making a protein. All right, this is a very basic introduction to the nucleic acids. I'm more interested that you understand some of the structure, comparing DNA and RNA, describing the structure of a nucleotide, and understanding that base pairing and understanding how to determine the complementary sequence, either DNA or RNA, because remember, there will be differences there.